Welcome back. I can't say that you look all rejuvenated and ready to go. If you're like me, you're probably in worse shape than when you left. Today we will have more of a regular class session. We don't really have anything to do regarding lab. This week is your lab presentations. Dr. Morgan's also going to go over the lab quiz with you, and so you will get that back today. Did you want to say anything about lab this week, presentations, etc.?
We'll go over the lab quiz, and I will give you about 10 or 15 minutes to talk with your lab partner to get your act together for your presentation. It's kind of tough on Monday and the Tuesday groups to be as prepared as you might like to be prepared to present. You'll still be able to finish on time by doing that. So, those of you who still need to do some computer work, it's okay if you want to come in during the lab period to do computer work because we won't be using the computers, but I won't necessarily be able to help you.
Any guidance for them in terms of how long typically a presentation has been taking, or?
What I'm figuring on is somewhere around 5 to 6 minutes for presentation and a couple minutes for questions. Oh, which reminds me, you either come with a computer with your program on your PowerPoint on it, or bring it on a jump drive, or email it to me and I'll load it on my computer. Right? If you have a Mac, we have the attachments for MacBook and MacBook Pro. I'm not sure we have the ones for MacBook Air.
Natalie's got several adapters downstairs. So.
Yeah.
So.
The easiest thing is to email it to me. I'll load it on my desktop, and it'll all be on one computer. That way, too, you can use the automatic advance that I have, plus my laser. I have a laser pointer for
If you weren't here Friday, you should talk to me about getting access to the video that we saw and the video questions, because those video questions you turn in, there will be something on the next exam about that video, and you will get your video questions back, the answers that you wrote down, to be able to use on that particular question on the exam. So it's available on YouTube, so you just really need the access to the video questions for that. You ready for the sprint? Things are pretty crazy this last 6 weeks. We've got an exam coming up, what, a couple weeks? Okay. Well, the material we're covering now, it's convenient and hopefully kind of well planned that we are going to be syncing up again with stuff that we will be doing in lab starting next week. So ultimately, we're going to be spending some time digging into the central dogma, and then also talk about regulation of gene expression. And one of the applications of regulation of gene expression, actually the regulation of gene expression gone bad, will be to talk about the molecular basis of cancer. And so there will be some tie-in to the video that we saw right before break. So what does the central dogma tell us? What is the central dogma? What does it say? You're going to be a tough crowd today, aren't you?
Their brain cells aren't firing.
No, they're not. What does it have to do with? DNA. Good. Central dogma says that DNA is transcribed into RNA, and then RNA is translated into protein. Protein is really the working stuff of the cell. Particularly important in terms of functioning proteins are enzymes, membrane transporters, contractile proteins. Really a lot of the stuff that a cell does and what makes the cell a cell and gives it its particular characteristics are the proteins that it produces. That information is encoded into the structure of DNA molecules, but during this process of getting from instructions to final product, there is some manipulations that the cell goes through to convert one type of nucleic acid into another, and then the information in this nucleic acid becomes eventually assembled into a functioning polypeptide chain, which does some action in the cell. We've known about the central dogma for quite some time. However, it's really instructive and important, I think, to kind of go through some of the history of our understanding of how we figured out this particular relationship. And the place we're going to begin with is looking at the structure of DNA. This is information you should have already had. Many of you probably had this in middle school even, where you've learned some of the basics about DNA, and so we're not going to spend a lot of time belaboring these points. That's what your reading is going to be for, but I want to give you some historical context about how this all fits together. So, where to begin? Well, let's start out with chromosomes right now, okay? Chromosomes have really been something that scientists have been interested in for a long period of time, since probably at least the 1950s. What's so special about chromosomes? Why would scientists be interested in these things called chromosomes? What do you think about when you think about chromosomes? What's it? Okay, they somehow store genetic information. So we've got these things within our cells. Where in the cells are chromosomes located? They're in the nucleus, right? And these particular things somehow encode genetic information. Well, we now know how they encode genetic information. Somehow the structure of chromosomes contains information that specifies the primary structure of polypeptide chains. Y'all remember what primary structure is? Primary structure is the kind and order of amino acids that are present in that polypeptide chain. And which amino acids you have influences how that polypeptide chain folds up. And what is how that polypeptide chain folds up determined? What is structure determined? Function. What it does. Okay? So somehow information is encoded into these molecules or these units of inheritance that we call chromosomes. And so that's one important reason that we're interested, is that somehow they store genetic information. How does a molecule store genetic information? Another thing that's interesting about chromosomes, and that is kind of puzzling, is chromosomes have to be able to replicate prior to both mitosis and meiosis. How many chromosomes are present in your cells? Kind of a trick question. Most of your cells have 46 chromosomes. Which cells in your body don't have 46 chromosomes? The gametes, okay, the eggs and the sperm. How many do they have? 23, half that number of chromosomes, okay? So the process of mitosis gives us whatever, two cells with the same chromosome content as the original parent cell had. So if your skin cells start dividing, okay, your skin cells are constantly dividing because you're constantly losing skin cells. They die and they shed off. So when a skin cell divides by mitosis, if the parent cell had 46 chromosomes, the two daughter cells have 46 chromosomes, right? Well, the only way that can happen is if prior to that division, the chromosomes duplicate. And they have to duplicate accurately, okay? Would it really be good for duplication of chromosomes to occur so that one daughter cell got 25 and one got 27? Well, that'd be really bad. How about 45 and 42? That's not good either, okay? We need to have each daughter cell have the same number and kinds of chromosomes as the parental cell did. And having too few chromosomes or too many chromosomes can cause abnormalities in those cells. Did you talk last semester about chromosomal abnormalities at all, and non-disjunction and getting too many chromosomes of one type or another?
Just touched on a little bit when it came to gender determination.
Okay. So this process of chromosomes replicating prior to both mitosis and meiosis is really critical. Well, how does a molecule precisely replicate itself? That's an interesting question. Well, these two questions of function are directly related to structure. So one of the places we need to begin in our discussion of the central dogma is, what are chromosomes made of? Well, we've already said one thing chromosomes are made of. They're made up of DNA. But chromosomes contain another type of biological molecule as well. What other biological molecule do they have? One of three chance. Carbohydrate, lipid, protein, right? Chromosomes are molecules that are made up of both DNA and protein. Prior to 1953, what did we know about chromosomes? Does anyone know the significance of 1953? In 1953, something really important happened in cell biology. A paper was published that really changed the whole future of biology. Who wrote that paper? That particular paper described the double helical structure of DNA molecules. Who's credited with that? Watson and Crick. You've all heard of Watson and Crick before, right? Has anybody not heard of Watson and Crick? Yeah. One of the threads that I'm going to include in our discussion of this central dogma idea, and particularly the structure of DNA, is that there were other individuals who should have been recognized and were not. How many of you have heard of someone by the name of Franklin? Okay, Patty raised her hand. A couple people have. Rosalind Franklin got gypped, basically. Watson and Crick received the Nobel Prize for their discovery of the structure of DNA. Rosalind Franklin should have been credited as well. They used a lot of her data, and we'll talk about which of her data they used. But she wasn't recognized until long after her death. How come? She was a woman. In 1953, women in science were just not that common. And the idea is that, women were second-class citizens as far as science was concerned. And so she has an interesting story that weaves through this saga of Watson and Crick and the important discovery that they made. So what was significant about 1953? The publication of Watson and Crick's paper about the structure of DNA. Maybe we'll go here, maybe we won't. Okay, so prior to 1953, what did we know? We knew that chromosomes were made out of DNA and protein. What we didn't know prior to 1953 is which of these two molecules contain the genetic information. You're familiar with these two terms from last semester, correct? Genotype and phenotype. What does genotype refer to? The genetic makeup of an individual, which A, G's, T's, and C's you have in your chromosomes. Okay, what does phenotype refer to? The traits, okay. So genotype really is a reflection of your DNA and what you have in DNA and what your sequence of DNA is. What is phenotype a reflection of? Well, really, what proteins you have, and oftentimes what enzymes you're producing. So we can frame the central dogma in kind of another way. Instead of saying DNA, RNA, protein, say, how does genotype determine phenotype? How does the fact that you have this particular genotype mean that you have brown eyes, for example? Well, it all has to do with the fact that DNA is encoding the genetic information to make proteins. At least now we know that it's DNA. But prior to 1953, we really didn't know which component of chromosomes actually contained the genetic information. Was it DNA or was it protein? Most researchers felt that protein was the most logical candidate for the storage function of chromosomes, for storing genetic information. Anybody have any idea why the smart money was kind of on proteins as being the part of the molecule that actually stored information? What do you know about proteins? What are they made of? They're made of amino acids. How many amino acids are there? There are 20 common amino acids. What's DNA made up of? What are the monomers that make up nucleic acids? Nucleotides. How many different kinds of nucleotides are there? There's 4, right? So if we've got something that has 4 building blocks and something that has 20 building blocks, which one would probably be a more robust storer of genetic information? Probably proteins, right? And does genetic information have to be pretty robust? Yeah, if you think about it, I mean, all the information to make your cells encoded into 46 of these things we call chromosomes, that's incredible. And so at the time, the smart money was on protein being the portion of the chromosomes that stored the genetic information. Well, there were a series of experiments that were done beginning in 1928 and up through the 1950s that were really directed at this question, what is the genetic information? Is the genetic information DNA or is it proteins? We knew it resided in the chromosomes, but chromosomes have both DNA and protein. So which one actually contained the genetic information? Again, most people felt it was the protein portion, and that the DNA part of the chromosome probably just provided some kind of structural backbone or something, if you like, but it really wasn't the storage portion of genetic information. It was protein, most people thought. So here are some people that you're going to learn about. Griffith, Avery McLeod and McCarty, and Hershey and Chase. During this 24-year period of time, some quite elegant experiments were done, and the conclusion from these particular experiments was that DNA was indeed the portion of the chromosome that encoded the genetic information. So let's take a look at some of these experiments and see how they contributed to that particular insight of DNA being the genetic information. Once we knew that DNA was the genetic information, then to answer these questions about how it stored information, we had to know something about the structure of DNA. So this realization that DNA was genetic information ultimately led to this quest to find out, what does this DNA molecule look like? What is its structure? Because understanding its structure could lead us hopefully to understanding questions of function, like how it stores genetic information, how it replicates. Word association. You should associate the research done by Griffith in 1928, and don't worry about the absolute value of these particular dates. Again, I put dates in there to give you kind of a historical context. Okay. What you should associate with Griffith's work is the discovery of a process called transformation. And that's going to be relevant to what work we're going to be doing in lab starting next week. Because we're going to do a whole bunch of experiments where we're going to transform an organism. What does transformation involve? It involves changing the phenotype of an organism. How do we change the phenotype of organisms? We give them some new DNA they didn't have before. We give them some new genes. So one of the transformation experiments that we're going to do is we're going to make glowing bacteria. And some of you might have even done this in high school. It's a pretty common experiment. We're going to then follow this up by doing a more detailed transformation experiment the last 3 weeks of lab. And again, we're going to change the phenotype of another organism. So how is transformation discovered and what's it involve? Griffith's work in 1928 dealt with a disease. And the disease was pneumonia. What's characteristic of pneumonia? What is pneumonia? Has anybody ever had pneumonia? Hassan, what happened to you when you had pneumonia?
Just constantly coughing, it's hard to breathe.
Coughing, hard to breathe. Why are you coughing? Why is it hard to breathe? You accumulate fluid in your lungs, okay? Really, pneumonia is the result of a process called inflammation. What can cause pneumonia? What can cause fluid to accumulate in your lungs? Do you know what the cause of yours was, Austin?
What?
Do you know what the cause of your pneumonia was?
No, I don't know. I was really, really young. I had
Okay.
to get on a ventilator and everything.
Okay. So what can cause the inflammation of your lungs? And that accumulation of fluid.
Bacterial infection.
There are bacterial forms of pneumonia. And how can we treat bacterial pneumonia? Antibiotics, right? There are also viral pneumonias. Do they respond to antibiotics? Nope. There's another type of pneumonia that can occur. Called aspiration pneumonia. Do you know what that is? Brendan, do you know what that is? That's when you inhale things into your lungs, like food particles, for example. I have a good friend from college who has real lung issues. He has trouble swallowing, and he frequently gets aspiration pneumonia. You're not supposed to inhale food into your lungs, but he does, and that causes inflammation. Griffith was working with bacterial pneumonia. And the species of bacteria he was working with was a bacterium called Streptococcus pneumonia. Streptococcus are little circular-shaped bacteria, oftentimes found in hairs. These forms of bacteria can be found in different types or different strains. And he worked with 2 different strains of Streptococcus pneumonia. One form was virulent, and one form was avirulent. What's virulent mean? You know? Good or bad? Bad. Okay. These are highly contagious, dangerous organisms. If a mouse is injected with a virulent strain of Streptococcus pneumoniae, usually it will die. What does avirulent mean, you know? Anytime you see that prefix a before a word, it typically means without. So these are non-virulent bacteria. If you inject a mouse with avirulent forms of Streptococcus pneumonia, they'll live, okay? The bacteria won't kill them. They won't cause pneumonia. Well, what was the difference between these 2 strains? Well, interestingly, if you plate these bacteria out on agar plates, and you look at the colonies that grow on the plates, these particular virulent bacteria, or the virulent strain, forms these nice circular colonies that have kind of a smooth, glistening appearance. And they're called the smooth strain of bacteria. If you plate out those avirulent bacteria, they have kind of a rough, irregular appearance. So there was something physically different between these 2 strains of bacteria, one which was lethal, the smooth strain, and one which was non-lethal, the rough stain. And some research was done. And as it turns out, these smooth bacteria had a unique structural feature called a capsule. So the encapsulated bacteria had this little halo-like structure that surrounded the bacterial cell. So this thing's the capsule right here. The rough forms didn't have the capsule. What's going on here? Why did having this capsule make you deadly? Why did lacking that capsule make you not deadly? Well, I suppose you could argue, well, maybe there's something in the capsule that's causing the pneumonia. And that was tested, and that wasn't the case. It wasn't the capsule itself. It was the bacterial cell that was causing the pneumonia. What this capsule actually did is it protected the bacterial cell. When you injected these bacteria into a mouse, normally the immune system of the mouse should be mobilized and attack that particular bacteria and destroy it before it can start multiplying. The mechanism that the immune system uses is phagocytosis. White blood cells should have gobbled those bacteria up. But the presence of this capsule protected them from that process. So they continued to live and thrive. And as they did, they caused inflammation. What about these cells without a capsule? They weren't protected. So when you injected them into the mouse, phagocytosis occurred. The cells got killed, and therefore they didn't cause pneumonia. Okay. Some further tests determined that the ability to produce the capsule was a genetic trait. So these virulent bacteria had some genes that allowed them to produce a capsule. And that's why they were deadly. These cells were mutants. They had some defects in their genes for making a capsule. They didn't make that protective structure, and therefore they were eliminated by the immune system. So when Griffith studied pneumonia, he really was looking at different strains that had different genes associated with them. And the genotypes of these two organisms determined their phenotype, their ability to kill or to not kill the mouse cells that they were exposed to. So, here's what Griffith did. He did one experiment where he took some living smooth cells, and here you can see the two cells, the two streptococcal cells surrounded by this capsule. He injected them into mice, and the mice died. Okay? So these were virulent bacteria. They were multiplying, causing pneumonia. Pneumonia was killing the mice. He did another experiment where he took the other strain, the living rough cells. They lacked the capsule, injected them into the mouse. Mouse didn't care. Why? Because the immune system was eliminating those cells before they could multiply. Then he took some heat-killed smooth cells and he injected those into the mouse. Mouse was healthy. Well, it makes sense because what kills the mice is these cells multiply. If you're injecting dead cells, those cells aren't going to multiply and therefore they're not going to cause pneumonia. But here's where he did something very interesting. He then took a mixture of heat-killed smooth cells, so these were dead, and mixed them with living rough cells. Heat-killed smooth cells by themselves are avirulent. The mouse doesn't die. The rough cells are avirulent. The mouse doesn't die. But mixing those two cells together results in mouse death. And furthermore, if you do an autopsy on these mice, you'll find their lungs are loaded with the smooth form of Streptococcus bacteria. That's why they die from pneumonia. So how can mixing one avirulent form with another avirulent form give us a virulent form? Well, there were a couple of possibilities. One was that somehow mixing these dead cells with these living cells caused the dead cells to come alive. It's a possibility, but it didn't make a lot of sense. Okay? There really wasn't any evidence that life could be resuscitated just by mixing it with another bacteria. What seemed to make the most sense was something was being released from these living cells, from these cells, these dead cells, which was converting these living cells into the virulent form. What might that be? DNA or genes that told cells how to make capsules. So these dead cells were releasing DNA. That DNA was being taken up by these living cells, and these cells were becoming transformed into the virulent form. Their phenotype was being changed. Griffith kind of stopped here. Didn't really take this any further. Did this particular experiment in and of itself answer the question, what's the genetic information? No, it really didn't. All we were able to deduce from this particular experiment was some genetic information from these dead cells was transforming these living cells. But we don't know what it was. Griffith never did identify the transforming substance. In 1944, Avery, McLeod, and McCarty recreated this experiment with some twists. In this particular experiment right here, they did take avirulent rough bacteria, but they didn't mix them with whole killed cells. They actually did 4 experiments. In one experiment, they mixed the avirulent bacteria with carbohydrate isolated from these killed smooth cells. And then they injected that combination into the mouse. What happened when they mixed carbohydrate from these smooth cells with the living rough cells? Mouse survived. They repeated the experiment, and this time they mixed the lipid from these cells with these avirulent cells. Injected those into the mouse. It lived. Then they tried protein. Took the proteins out of these cells, mixed them with these. The mouse still lived. It was only when they took the DNA portion of these killed cells and mixed it with these living cells that transformation occurred and the mouse died. So Avery, McLeod, and McCarty were able to definitively demonstrate that DNA was the substance that caused genetic transformation. And we knew that that particular substance contained the genetic information for how to make a capsule. Well, that should have been pretty compelling evidence by itself. As it turns out, another experiment we'll talk about tomorrow was done later that ultimately reinforced Avery, McLeod, and McCarty's conclusions and further demonstrated that DNA was the genetic information. Make sure you've got a front and a back. Looks like the master got. So there should be 11 questions. Oh, well, I mean, I think both possibilities would be considered, this idea of reanimating life, you know.
Well, then, the Avery McLeod Carter experiment, you would think, must have control when you took the DNA from this.
Oh, I'm sure it is, yeah. I'm sure it is.
I have to find that paper. These are so elegant in their simplicity.
Yeah.
Or at least the way we teach them.